Welcome back to the Deep Dive. Today we're really tearing into the essentials, the absolute foundations of microbiology.
That's right.
If you're heading into healthcare, or honestly, if you just want to understand the microscopic world around us and, frankly, inside us, this is where it all starts.
It really
We're
is.
aiming to pull out the core concepts, microbes, disease, their impact. It's crucial stuff.
Definitely. Our mission really is to quickly get you up to speed on the history, how we discovered this hidden world, and the big ideas, the theories that changed everything.
And the classification systems too, right? How we make sense of it all.
Exactly. And how it all applies today in the clinic, in our lives. This isn't just dusty history. It's the why behind so much modern medicine.
Okay, so let's start with that history because, well, for most of human existence, we were completely in the dark.
Utterly blind, yeah.
We know diseases like cholera, tetanus, polio. They've been around since maybe 4000 BCE. But knowing what caused them.
Nope. Took a long, long time.
Our sources highlight that famous story, London, mid-1800s, John Snow and the cholera outbreak.
Ah, yes. The Broad Street pump. Classic.
His big move was just removing the pump handle. People stopped getting water there, and the epidemic slowed down.
It was incredibly effective public health. But, as the sources say, kind of serendipitous, he didn't know about Vibrio cholerae, the actual bacterium.
He just saw the pattern, drink the water, get sick, stop the water.
Stop the sickness. It was brilliant observation, but the how and why needed technology. It needed us to actually see the culprits.
Which brings us to the microscope pioneers in the 17th century. Two key figures pop up immediately.
Right. First, Robert Hooke.
He had a compound microscope, maybe got, 20 or 30 times magnification?
Yeah.
Enough to see larger structures, cell walls, and cork.
Important first step. But
Yeah.
then came Antony van Leeuwenhoek.
The game changer.
Absolutely. He was apparently an amazing lens grinder, pushed single lens microscopes way further, maybe up to 200 times magnification.
And that jump was enough.
It was the jump. It let him be the first person to actually see and describe single-celled organisms, bacteria, protozoa. He called them animalcules.
Little animals. That's why he's called the father of modern microbiology, isn't it? He literally opened our eyes to that world.
Precisely. He made the unseen seen.
Okay, so Leeuwenhoek saw something. But now we need to see much more, right? Internal structures, how things move, surface details.
Exactly. We need a whole toolkit of microscopes now, each designed for a different task.
So what's in the modern toolkit?
Well, broadly, you've got light microscopes and electron microscopes. Your standard workhorse is the compound light microscope. Uses visible light, multiple lenses.
Ocular and objective lenses, yeah.
Right. Gets you up to about 1,000 times magnification.
And the most common type is Brayfield.
That's the one. Bright background. But the catch is, most cells are basically transparent.
So you usually have to kill them and stain them to see anything clearly.
Which isn't great if you want to see something alive and moving.
Right. So what do you use then?
That's where something like a dark field microscope comes in. It's clever, it blocks the direct light.
How does that help?
Only light that hits the specimen and scatters up towards your eye gets through. So you see a bright, living organism, maybe bacteria swimming, or a spirochete twisting against a pitch-black background. Really useful for unstained living samples.
Okay. And what if you want to see inside a living transparent cell? Like watching it divide or seeing stuff move around in the cytoplasm.
Ah, then you want phase contrast. This one uses the physics of light bending.
How so?
Light bends differently as it passes through parts of the cell with different densities. The scope enhances these tiny differences in the light waves, turning density variations into visible contrast.
So you can see internal structures without staining while it's alive.
Exactly. You can watch things like cytoplasmic streaming, cell division, all in real time. It's fantastic for that.
Then there's fluorescence microscopy.
Yeah.
Sounds different.
It uses UV light. Some things naturally fluoresce, like chlorophyll, but more often you tag something specific, like an antibody for a certain pathogen, with a fluorescent dye.
Ah, so it glows under UV light if the target is present. For diagnostics.
Precisely. It's a very powerful diagnostic tool. If it glows, you know your target microbe or molecule is there.
Okay, that covers light microscopy. But for really high magnification and detail, electron microscopes, right? EMs.
Right. These are the powerhouses. They use beams of electrons, not light, and magnetic fields, not glass lenses. Much, much higher resolution.
And there are different types of EMs, too.
Two main types. First, the transmission electron microscope, or TEM. It shoots electrons through a super thin slice of the specimen.
Like an X-ray, but with electrons.
Sort of, yeah. It gives you an incredibly detailed 2D image of the internal structures. Think like a million times magnification. You see the organelles, the membranes.
Yeah, okay, and the other type?
That's the scanning electron microscope, or SEM. This one scans the electron beam across the surface of the specimen.
So not looking through it, but at its shape.
Exactly. It builds up a high-resolution 3D-like image of the surface texture. TEM shows you the inside, SEM shows you the outside in detail.
Got it. And I think the sources mentioned one more. Atomic force microscope, AFM.
Oh yeah, AFM is really cool. It doesn't even use beams in the same way. It has this tiny, sharp stylus.
Like a record player needle?
Kind of, but unbelievably fine. It physically taps or drags across the surface, tracing the contours. It builds a topographical map, almost like feeling the shape at an atomic level. And you don't usually need to coat the sample or put it in a vacuum.
Incredible technology.
Yeah.
And having these tools, this ability to finally see microbes, was key to settling that huge debate, wasn't it? Spontaneous generation.
Oh, absolutely critical. The idea of abiogenesis life just popping out of non-living stuff.
Like maggots from rotting meat or mold on old bread. Seems obvious to us now, but it was a real theory.
A dominant theory for centuries. Francesco Redi made the first good challenge back in 1668.
With the meat in jars experiment.
Yeah. Meat in open jars got maggots. Meat in sealed jars or jars covered with gauze didn't. Showed the maggots came from flies, not the meat itself.
But that didn't settle for microbes, did it? There's that whole Needham versus Spallanzani thing with boiling broth.
Right. Needham boiled broth, sealed it, and saw growth. Claimed it proved spontaneous generation. Spallanzani boiled it longer, sealed it better, and saw no growth.
So Spallanzani disproved it.
He made a strong case, but critics argued he'd destroyed some vital force in the air, or that life needed air. The debate lingered.
Until Pasteur.
Until Pasteur 1861. His experiment was the definitive answer.
The swan-necked flasks. Explain those again.
Okay, so he put nutrient broth in these flasks with long, curved necks like a swan's neck. He boiled the broth to sterilize it.
Killing any existing microbes.
Right. Now, the crucial part. The S-bend in the neck let air move freely in and out, so the vital force argument was addressed.
But the curve trapped dust and microbes from the air, right?
Exactly. Gravity pulled them down into the bend. They couldn't reach the sterile broth, and the broth stayed sterile, clear, indefinitely. Until Pasteur tilted the flask, letting the sterile broth wash back into the dusty neck bend. Then, microbial growth happened almost immediately.
Proving life only comes from pre-existing life. Omne vivum ex vivo.
Precisely. It slammed the door shut on spontaneous generation for good.
And with that settled, the focus could shift squarely onto the germ theory of disease, which led pretty quickly to practical changes like asepsis.
Yes, the idea of preventing contamination. Ignaz Semmelweis in Vienna, mid-1800s, a tragic hero, really.
What did he do?
He noticed women were dying of childbed fever at alarming rates in the ward where doctors and students came straight from doing autopsies.
Without washing their hands. Ugh.
Yep. In the other ward, attended only by midwives, the death rate was way lower. Semmelweis made the doctors start washing their hands in a chlorine solution between autopsy and maternity ward.
And the death rate
plummeted. He proved the connection, but sadly, the medical establishment largely rejected his ideas at the time.
But the idea caught on, eventually, thanks partly to Joseph Lister.
Yes, Lister took it further. Inspired by Pasteur's work on fermentation and putrefaction being caused by microbes, he started using carbolic acid phenol as an antiseptic during surgery.
On wounds, dressings, instruments, even spraying it in the air.
Right. He pioneered modern aseptic surgical techniques, drastically reducing post-operative infections.
So, Semmelweis and Lister showed preventing germ transmission worked. But who actually proved that specific germs cause specific diseases? That sounds like Robert Koch.
That is absolutely Robert Koch. His work, especially on anthrax, was foundational. He didn't just suspect a microbe, he proved it using a rigorous method.
Koch's postulates. Every healthcare student has to learn these.
They do. They're the 4 criteria you need to meet to definitively link a microbe to a disease. Can we run through them quickly?
Yeah, let's do it. Postulate 1.
The suspected microorganism must be found in every case of the disease and be absent from healthy individuals.
Makes sense.
Number 2.
The microorganism must be isolated from the diseased host and grown in pure culture in the laboratory, away from the host.
Okay, so you've got it growing by itself. Number 3.
When that pure culture is inoculated into a healthy, susceptible host, it must cause the same disease.
You have to reproduce the disease. And the final one, number 4.
You have to be able to re-isolate the same microorganism from that newly infected experimental host.
Closing the loop. Find it, grow it, cause it, find it again.
That's the logic. Now, we know today they don't apply perfectly to everything. Viruses are hard to culture. Some diseases have asymptomatic carriers. But the principle, the rigorous approach, is still core to microbiology and epidemiology.
Okay, so we can identify the culprits. How do we organize this vast world of microbes? Classification time.
Right. Let's start with the time scale. Life's been around a long time. Prokaryotes, the simpler cells, showed up maybe 3.5 to 4 billion years ago.
We see evidence in things like stromatolites, those layered rock formations made by ancient microbes.
Exactly. Eukaryotes, the cells with the nucleus and internal compartments, like our cells, came much later, maybe 2.2 billion years ago.
So the fundamental split is prokaryote versus eukaryote.
That's the biggest structural difference, yes. Prokaryotes, bacteria and archaea, lack that membrane-bound nucleus and organelles. Eukaryotes, algae, fungi, protozoa, plants, animals have them.
And we now use the 3-domain system based on genetics, specifically ribosomal RNA sequencing.
Correct. Domain bacteria, domain archaea, and domain eukarya.
Archaea are the really interesting ones, often found in crazy environments, right? Hot springs, glaciers, super salty water.
Yeah, the extremophiles. Genetically distinct from bacteria, even though they look similar under a basic microscope. They tell us a lot about the limits of life.
But not everything infectious fits neatly into those cellular domains. We have the non-cellular agents.
Right. Viruses are the big ones here. They're not cells. They're basically just genetic material, DNA or RNA, wrapped in a protein coat.
And they can't reproduce on their own.
Nope. Metabolically inert. They have to hijack a host cell's machinery to make copies of themselves. Obligate intracellular parasites.
Then things get even weirder with prions.
Oh, prions are mind-bending. They are infectious proteins.
No DNA, no RNA, just protein.
Just protein. A misfolded version of a normal protein found in the brain. This misfolded prion bumps into normal proteins and causes them to misfold too.
Like a chain reaction of bad folding.
Exactly. It leads to devastating neurodegenerative diseases, mad cow disease in cattle, Creutzfeldt-Jakob disease, CJD, in humans. The idea that a protein alone could be infectious was revolutionary, even heretical at first.
And even smaller than viruses, you mentioned viroids?
Yeah, mostly known as plant pathogens. They're incredibly simple, just a tiny naked circle of single-stranded RNA, no protein coat, nothing else, still manages to cause disease in plants.
Amazing diversity. So, to keep it all straight, we use taxonomy.
The formal system for naming and classifying, we still use the hierarchical system largely developed by Linnaeus.
Domain, kingdom, phylum, class, order, family, genus, species. Did I get that right?
You got it, and the naming convention is key. The binomial nomenclature.
Genus capitalized, species lowercase, italicized, or underlined? Like Escherichia coli, or E. coli for short.
Crucial for clear communication worldwide. Everyone knows exactly which organism you mean.
Okay, moving beyond just naming them, how do microbes live and interact? You mentioned biofilms.
Ah, biofilms. Super important, especially in healthcare. These aren't just free-floating microbes. They form organized communities attached to surfaces.
Like on teeth as plaque, or inside catheters, or on implants?
Exactly. The cells stick together and produce this slimy, protective extracellular matrix around themselves. Think of it like a microbial city shielded by walls.
And that matrix makes them hard to treat.
Incredibly hard. It protects them from antibiotics, disinfectants, even the host's immune system. Biofilms are a major challenge in persistent infections.
Within these communities, or just generally, microbes have different kinds of relationships, right?
Yes, ecological relationships. You've got mutualism, where both species benefit. Think E. coli in our gut, making vitamins for us while we give it a home.
Commensalism.
One benefits, the other is unaffected. Maybe one microbe eats the waste products of another.
Synergism.
Where two or more microbes work together to do something neither could do alone, like breaking down a complex compound. They depend on each other for that task.
And the obvious one, parasitism.
One benefits the parasite at the expense of the other, the host. That's basically the definition of a pathogen causing disease.
Which brings us to the microbes living on and in us normally, the normal flora. They aren't parasites.
Mostly not, no. In fact, they're usually beneficial, often mutualistic or commensal. They live on our skin, in our mouths, dominate our gut.
And they're important for health.
Absolutely vital. They help digest food, produce vitamins, and crucially, they protect us from pathogens.
How do they protect us?
By competitive exclusion. They take up space, use nutrients, sometimes produce substances that inhibit dangerous invaders. They're like our first line of microbial defense.
But it's important where they stay, right?
Yes.
Certain areas need to be sterile.
Yes, critical distinction. Blood, cerebrospinal fluid, CSF. Internal organs like the lungs or bladder, these should be sterile. If normal flora from your gut gets into your bloodstream or bladder, it can cause serious infection. They become opportunistic pathogens.
Okay, so when pathogens do get in and cause disease, how does it spread? Transmission.
Several routes. Direct contact, obviously.
Yeah.
Indirect contact via contaminated objects. Then there's airborne transmission through aerosols. Think coughing or sneezing.
Waterborne too, like the cholera example.
Definitely. Contaminated water supplies are a major source, especially after floods or in areas with poor sanitation, cholera, typhoid, dysentery.
And foodborne transmission.
Right. Contaminated food or water used in food prep, or sometimes toxins produced by microbes in the food.
So prevention still comes back to basics.
Largely, yes. Hand washing is huge. Cooking food thoroughly. Ensuring safe drinking water through purification, boiling, or chlorination. Basic hygiene practices rooted in the germ theory.
Finally, let's talk about how we use microbes deliberately. Applied microbiology.
Yeah, humans have been harnessing microbes for millennia, often without knowing it. Think food production.
Fermentation?
Exactly. Yeast like Saccharomyces cerevisiae for bread and alcoholic drinks, lactic acid bacteria for yogurt, sauerkraut, kimchi, cheese.
The source mentioned Propionibacterium shermanii for Swiss cheese.
That's the one. It produces the CO2 gas that makes the holes and contributes to that distinctive nutty flavor.
Cool. What about medicine? Pharmaceuticals?
Well, the discovery of antibiotics fundamentally changed medicine. That started with Fleming noticing Penicillium mold inhibiting bacteria that's antibiosis, one microbe killing another.
And now we harness that and even engineer microbes.
Yes. We search for natural antibiotics, and we use genetic engineering to make microbes produce specific drugs, hormones like insulin, vaccines. It's a huge industry.
And using microbes for cleanup, bioremediation.
Also very important, using naturally occurring or engineered microbes to break down pollutants. Think oil-eating bacteria after a spill, or the microbes essential for sewage treatment plants. They digest the waste.
They can even make fuel.
Bioconversion, yeah. Microbes can ferment biomass into ethanol or produce methane gas from landfill waste. Alternative energy sources.
And a newer field of microbial forensics.
Fascinating area. Using microbiology to trace the source of pathogens in things like bioterrorism investigations, remember the anthrax letters, or tracking outbreaks of foodborne illness, or even in cases of medical negligence. It's about finding the microbial fingerprint.
Wow, okay, so wrapping this all up, the journey of microbiology is incredible. From just wondering about animalcules
to understanding intricate genetic codes and complex ecosystems like biofilms.
The big theoretical leaps, disproving spontaneous generation, establishing the germ theory with Koch's postulates, they literally form the foundation of all modern healthcare and public health.
Couldn't agree more. And it highlights that microbes aren't just villains. They're essential recyclers in ecosystems, vital partners in our own health as normal flora, and powerful tools in applied science, food, medicine, environment.
Absolutely. Okay, let's leave our listeners with something to chew on. Here's a final thought for you.
Go for it.
We've talked about the power of antibiotics, stemming from these foundational principles. But microbes evolve fast. We're facing superbugs, MRSA, drug-resistant C. diff, resistant tuberculosis. We're seeing diseases like measles reemerge.
A serious concern.
So the question is, when our current frontline drugs, like maybe the common cephalosporin antibiotics, start failing more widely, how will the fundamental principles we discussed today, understanding transmission, asepsis, microbial physiology, maybe even harnessing other microbes, guide us in developing the next generation of defenses? How do we stay ahead in this arms race?
That is the question for the future of infectious disease control. It forces us back to these core ideas.
Something to think about. Thanks for joining us on this deep dive. We'll catch you next time.